Very good morning, students. I hope you are doing good and well. Today, we are going to discuss the semi conservative method of DNA replication, that is, the Mesalsan Stahl experiment of DNA replications. Initially, this Mesalsan Stahl experiments of replication, it means the replication of DNA. It wasn't published in PNAS, Proceeding of Natural Academy of Sciences USA in the year of 1958. This concept helped the cement of the concept of double helix of Watson Crick. The Mesalsan is a graduate student and Stahl is a postdoctoral researcher from California Institute of Technology. They gave a validity to the model that how two intertwined and tangled strands of helix could physically code for the material of inheritance. These Mesalsan talk experiments have the approval of Watson Crick model of DNA models. Okay. In this topic, these are the three major components. One is the in the introduction, then mode of DNA replication, as well as the Mesalsan stock experiments. And briefly, we are going to discuss. The introduction part, the Matthew Mesalsan as well as the Franklin Stahl, in the year of 1958, performed the most beautiful experiment in the biology, which supported the hypothesis proposed by Watson Crick that DNA replication was semi-conservative. What is DNA replications? We already knows. The DNA replication is meant to produce the new molecules that have the same base sequences. Is it right? In the right side, we are looking the cell cycle. Exactly in the cell cycle, during the S phase, stationary phase, the DNA replication could take place. Okay, the mode of DNA replication. The DNA replications have the three possibilities. One is semi-conservative replications. It means the parental DNA, when it go for F1 progeny, the parental double-stranded DNA is separated into single stranded, and each single stranded parental DNA, one new strands of DNA will be formed. That is the semi-conservative. The second one is conservative. It means the would preserve the original molecules. It means the double-stranded DNA during the F1 progeny, it stays a double-stranded parental DNA in complementary double-stranded new DNA will be formed. That is conservative. Third one is dispersive replications. Here, two molecules will form with the help of parental DNA, and those DNA will be interspiced along with each strands. That is dispersive. In the picture, you will clearly observe what is the difference between semi-conservative, conservative, as well as the dispersive. Here we are going to discuss the semi-conservative replication of DNA through Mesalsan-Stahl experimental model. Okay, what they are done it. The Mesalsan and Stahl experiments, they conducted the DNA replication in E. coli bacteria. E. coli is a model system. Okay, the beginning, the E. coli medium is grown. Generally, we used to grow in our laboratory E. coli in a nutrient broth. Is it right? Yes. In the nutrient broth, they used to add isotope. It means heavy isotope of nitrogen like N15. Then bacteria took up the nitrogen and used to synthesize new biological molecules including DNA. Then after many generation growing in N15 medium, the nitrogen bases of the bacteria's DNA will be labeled as heavy N15. Okay, this is the first step. Then later, the same bacteria were switched to the medium containing light N15 isotope. So allowed to grow again the several generations. So what will happen? DNA made after switch would have to be made up of N14. As this would have been only the nitrogen is available for the DNA synthesis. In that, later on that, we have to collect the bacterial sample for the each generations and extract and purify the DNA and measure the density of the DNA gradient using density gradient centrifugations. Here we are used two isotopes. One is heavy N15, second one is light, it means low density N14. Now in the density gradient centrifugation, we used to add cesium chloride solution, okay, from on top of this finning tube. Then what will happen when the density gradient centrifugation is happened? When it is N15 for initially in between 0 hours, first we are what we done it, the E. coli is to grow under N15, it means heavy isotope medium. There we are centrifuged, what will happen? It means heavy, so 100% of the progeny is N15. Okay, then later what will happen? We added N14 light, then again we are growing. Then each and every generations, first progeny, second progeny means generally we used to know that bacterial growth is 24 hours. Okay, so each time intervals we used to collect the culture, then we have to be centrifuge and we have to get look into it, how much density is increased or decreased. So when we do it and when we are doing simultaneously simultaneous time intervals, 20 minutes, 40 minutes, 60 minutes, 80 minutes, what will happen? Then each generations the DNA density is varied. DNA density is varied. When it is N14 is always light, so it will be top. When it have both interfined, it means heavy N15, it's always in the bottom. When the DNA, bacterial DNA having both N14, N15, what will happen? It will stay in the middle. So it is the semi-conservative. Two parallel DNAs, when go for the replications in the semi-conservative case, what will happen? One parallel DNA, one complementary. One parallel, one complementary. So the middle origin intermediate type of DNA will be occur. So in E. coli, during this test, what we conclude? The Mesalsan Stahl demonstrated the DNA replication is be semi-conservative, meaning that it stands up the DNA molecule, serves as a template synthesis for the new DNA to the complementary strand. Okay, now you understand that. Okay, thank you very much.